Hi everyone! Wake up. My name's Claire, and so today I will be talking about using biochemical markers to investigate the hidden lives of planktivorous elasmobranchs. Basically, I chose the cute spotty fish. My base is in Mozambique, in Tofo. This is my office. It's really horrible. I work with the Megafauna Foundation, and I think some of you know my supervisor, Dr. Simon Pearce, the lovable Kiwi. I also work with the University of Southampton very recently, and working with the National Oceanography Centre specifically. So I'm pretty sure everybody knows what a whale shark is, but I'm just going to quickly go over some things you might not know. So, whale sharks are distributed in tropical and subtropical waters across the world. They're the biggest fish in the sea. They are traditionally known for their coastal aggregation behaviour. So there are certain hotspots around the world, but most of them I'll be talking about are in the Indian Ocean. Specifically Mozambique, Tanzania, Djibouti, and in fact there are more hotspots being discovered all the time as people, I guess, colonize the coast more. What's interesting about the Indian Ocean populations is that they pretty much all show size and sex segregation. So all of these coastal aggregation sites are actually predominantly made up of the young, immature males, so all the teenage boys that hang around at the beach. This is one of the other things about the whale sharks is that although we know them for their coastal aggregation behaviour, typically this is only for a few months of the year at each of these locations. So these actually pose a few problems for researchers. We are very, very unsure about what the behaviour of the mature adults is like, especially the females. We really don't see very many young sharks. I think very recently, in the last few weeks, we saw a shark that was about 2.5 meters long, and that is, I believe, from Andrea, who's been there for over 10 years, that is the smallest whale shark that's ever been seen there in the last 10 years. So, as they're born at about 50 centimeters long, we really are missing both the low and the high end of the demographic here. We also, because they're only really around these coastal sites between, I guess, 3 and 6 months of the year, depending on where you are, we have no idea where they go after that or what they do. They're very, very large, they're very highly migratory, and this means that we really are not getting the full picture of what these guys are doing. So, we have tried to take a different approach, and using all these new ecological tools, we're sort of using the are what you eat principle. So this is really how fatty acid analysis and stable isotope analysis works. I'll be covering both very briefly, just to give you an idea. Hopefully, for my PhD project, I'll be trying to combine the two to get a bit of a holistic view of what's going on. So, like I said, they both work on the principle of you are what you eat. So, what the predators are actually eating affects what goes into their tissues. So, you can, in terms of fatty acids, it works very well. You can actually get sometimes quite a sort of small-scale view of what they're eating. So predators cannot synthesize certain fatty acids themselves, and therefore we know that they come directly from their diet. And by comparing the profiles of the prey and the predator, you can get quite a good idea of either exactly what they're eating or a very good breakdown of what they're eating. In terms of stable isotopes, I think Johannes touched on this as well, but I'll go over it again. You can look, by looking at different stable isotopes, classically in marine ecology we use carbon and nitrogen isotopes. These vary fairly predictably over latitudes, so ocean basin scales. They vary over depth, so the surface and sort of mesopelagic zones will have different ratios. They vary over ecosystems, so you can look into diet shifts as well, and specifically nitrogen varies over trophic levels fairly predictably as well. So you can start to get a really good picture about what they're eating and where they're eating, which is the idea. The benefits of using stable isotopes and fatty acids are that you can get a really large sample size very, very quickly For example, for the whale sharks, I think last year we got 60 plus samples in a few months, and when you sort of compare that to the identified population, which is about 600, 650 at the moment, you're kind of getting towards something which is becoming an acceptable representation of your population. They're time integrated as well, so while using, it's usually better to use these in conjunction with necropsies and techniques like this. These sort of stomach content analysis, really produce a very specific snapshot of the diet of these individuals. So you're really just getting their last meal, which often doesn't tell you a great deal about what they're eating the rest of the time. For whale sharks specifically, we only really get stomach content analysis when the whale sharks get washed up somewhere. Quite a few of ours we've got from Jeremy, thanks. And this is because they're getting washed up on shore. Really what they've been eating has been the sort of surface zooplankton right at the coastal areas, and then we don't have an idea of what else they're eating as well. They're relatively inexpensive, so analysing them, I'm going to try and convert it to rand. So for one sample it costs about 140, 150 rand for a sample, and then when you compare this to the $4,000 you're spending on one tag, it starts to look really good. So another really good benefit about this, especially for whale sharks in particular, is that it's really minimally intrusive and it's non-lethal. So we don't, not like it happens very often, but we don't sort of get specimens from trawlers. We don't wait for opportunistic strandings, which don't happen very often. But also for species which are sort of vulnerable and protected, you really don't want to be killing them to do your research. So this is a good benefit as well. Unfortunately, there are drawbacks. Both stable isotope and fatty acid analysis, especially in marine ecology, is relatively new. There's still a lot of things we don't know, which means that interpretation of these results can be a little bit subjective, unfortunately. There are some things you do really have to consider. For stable isotopes, you have to know about tissue turnover rates, so you have to know what time scale you're working on. For example, you can imagine that the metabolic turnover rate for blood and liver tissues is a lot faster than muscle or skin. So you have to know what time scale you're looking at so that you can interpret your results correctly. Unfortunately, for tissue turnover rates, they're incredibly specific to species. So at the moment, I think the closest controlled feeding experiment that's been done to have a look at this was in leopard sharks by Kim et al. In 2012. So that's really the closest thing I have for my study, which isn't ideal, but you kind of have to work with what you have. For fatty acids, it's a similar story as well. You need to know things like synthesis and fractionation. So you need to know how these animals are processing these fatty acids once they get into their body, and then how they store them in different tissues as well. You can imagine the storage in muscle, the storage in skin. They're not going to store them exactly the same way in these lipid deposits. So you have to, again, have an idea of how they store which fatty acids where before you start making assumptions. And then, because this is a relatively new area for marine ecology and especially elasmobranchs, we really don't have very much baseline data. Stable isotopes have been traditionally used really in land-based research, and so for North America it's brilliant. You have this beautiful map with very well-defined lines, and you can pretty much tell to the mile where this bird has flown or where this marmot has been or whatever. Unfortunately, the ocean by nature is fluid. It makes these things a little bit more difficult. It's very, very changeable, and it's very, very hard to have very, very defined areas. So if a whale shark, for example, left Tofo, put on a jacket and went all the way down to Antarctica, we should be able to tell that, but it's very, very difficult at the moment because you don't have very, very good values across these latitudes to be able to tell these things exactly. There's also just a general lack of studies on elasmobranchs. It's getting a lot better. People are publishing more work on this, but it's considering things like urea retention and how that would affect the nitrogen levels in different tissues, and it's all these little things that you have to consider, especially for the sharks and rays, which we're still learning about at the moment. So the uses that I have here Johannes touched on this as well. The sort of carbon isotopes and nitrogen isotopes are traditionally used for this kind of research. Carbon is more where you've been eating, so it's set by the baseline levels at the base of the food chain, and it doesn't really change very much once you go up the trophic levels, so you can really kind of get a good idea about where these animals have been and what they've been eating there. The nitrogen is mostly for trophic level and sort of food web research, so you can, the nitrogen gets more enriched, so you get more of the heavy nitrogen 15 as you go up the food chain, so then you can start positioning animals and getting an idea of where they are in the food chain and where they are in their food web. You can, once you start knowing where these animals are going and what they're eating, you can then start to look into slightly more complicated things like habitat use. If they're spending more time in one area than another, this will show up in the fatty acids and stable isotopes because they'll be eating more in certain areas than others. You can look at residency. I mean, just because we don't see the whale sharks in the coastal areas, they might literally just be at, 2 k's off. We can't see them. They might be very resident and they're just next to us. We just can't see them. Or they might be just going straight off into the middle of the Indian Ocean, spending all their time there. We can start having a look at these values and seeing which one of these might be true. A slight aside to my project is hopefully going to have a look at niche partitioning. We're very lucky in Tofo because we're one of the only places in the world where we get whale sharks and both species of manta rays, the giant manta and the reef manta, all using the same habitat at the same time. And so something I'm going to try and look into is comparing these values across all the 3 species and see if there is sort of niche partitioning in this very small area. Obviously, everybody wants to look into the sex and ontogenetic effects, whether habitat use or you get dietary shifts as the size of the sharks changes. And another thing that I want to do is have a look at a bit of more of a worldwide analysis and step back and look at everybody else's populations of whale sharks as well as our own. So the way we do our sample collection is fairly straightforward and delightfully cheap again, hooray. So we just use a modified Hawaiian sling, so it's a sort of a spearfishing pole. You can kind of see the elastic on the end. And then we have sort of tips that we have a modified adapters for. They're about 10 centimetres long or so, and we aim pretty much the same place that everyone else uses to tag sharks, sort of just below the dorsal fin on either side. We try and go in between. I don't know if you can see the ridges on the whale shark. Those are actually quite hard. We try and aim between those. And just get basically a core of tissue, which for us is actually all connective and skin. We don't actually end up getting any red muscle, which is another problem, unfortunately. So this is relatively easy, and I would say I don't have any stats on this, but from personal observation, I'd go ahead and say about 90% of the time they don't even know you're there, let alone react. It's great, pretty but dumb, it's awesome. So these are the proposed locations I have, other than Tofo and Mozambique, which you can see a little spot at the bottom there. I'm working with several other researchers around the world and hoping to do some pretty worldwide collaborations with these guys as well. Because this tissue sampling is really easy and really cheap, it means that people are much easier to convince to get involved in this kind of thing. So as well as looking in the Indian Ocean, I'm hoping to have a look on the Pacific side as well. I think it's going pretty well so far. I think if I've got samples from Mozambique, Tanzania, Qatar and the Philippines, and that's probably, between the 3 of them, it's up to like 180 or so samples. So I'm really psyched because I think the last project that's looked into stable isotopes and whale sharks was Burrell outside of India, and I think his sample size was 9. So we're doing really well. I think that's plus Ryan's stuff, and we're doing really well, I think. So I'm quite glad this is going well so far. If anyone wants to give me any whale shark bits, again, that'd be great. Well, I'm also going to present now because I was hoping to present some initial results from my own data, but there's been a bit of a hiccup with the lab. This is one of these curveballs we were talking about. So I'm going to present very briefly some of the results by Chris Rona. I actually worked with him, as in I collected most of the samples for him for this initial study on whale sharks, looking specifically at fatty acid analysis. So the outline of his project was to look at both Mozambican and South African sharks. He collected the subdermal connective tissue in the same way that I described before. I think he had 24 individuals in the end. He collected surface zooplankton and sort of deeper zooplankton off the shelf break. He also had 5 stomach contents. Again, thank you, Jeremy, for some of those. He compared these to some already published fatty acid sort of results from potential and published prey species of the whale sharks. He also used a technique where he used mixed models, so he basically made up about 5 fake diets of the whale sharks and had different prey items in different proportions to see whether that would match up. It makes sense for these really large migratory species. They probably just don't eat one thing, so it makes sense to have a look at some combined species. Okay. Just the results really quickly. So the coastal zooplankton didn't match the fatty acid profiles of the sharks at all. They did, in the stomach contents, they did see some demersal zooplankton. So this is zooplankton that generally lives in the sort of sand and sediment during the day and then comes up at night time. This was backed up by some bacterial fatty acids that we found in the shark, so this may have been gotten through this detrital link here. The one dietary mix that seemed to group well with the whale sharks was Chris included all the prey that were within 40% similarity of the whale sharks for the initial analysis. And they also grouped together fairly well with some meso and bathypelagic plankton as well. This is just to show quickly that the sort of whale sharks are basically on that blob on the right, and then all the surface plankton is on the left. So clearly, even though whale sharks have been observed eating coastal zooplankton at the surface, it's not really their main dietary source. And this is a very scary graph that took me hours to understand. But basically, it was just to show that these are the whale sharks here. They really don't group well with any of these prey species on their own, but this orange dot in the middle was the dietary mix of all the prey species which were within 40% similarity here. So basically what that means is that because whale sharks are very large, they're very migratory, and they have work in a very patchy food environment in these tropical ecosystems. The whale sharks clearly forage during the day because we've seen it. It looks like they forage at night, and it looks like they eat these mystids or this zooplankton that sort of comes up from the sediment at night. And they do have links to both pelagic and deep water zooplankton as well. So they're they do eat or use several different ecosystems as well as these coastal ones. However, these samples were limited, the stomach contents were limited, so we can't really generalise. We only had samples from Mozambique and South Africa. So this is just really a good platform for me to do my work on top of this as well. However, this is very exciting news because whale sharks may actually play a part in some nutrient cycling, because they eat and spend time in so many of these different areas. They may actually contribute quite a great deal to this. So this is good stuff to work with. This is just a shopping list, a Christmas list, really. So as well as the fatty acid and stable isotope work, I really hope to do some acoustic tagging as well. So for the fatty acid and stable isotopes, you really don't need very much tissue, so if anyone happens to come across a whale shark, it would be really nice if you could give me a call. I'll be handing my card out shamelessly later. Also, there is a possibility of doing some ontogenetic or diet switching work, where I'll be using the whale shark vertebrae and actually taking samples from the centre all the way out to the side. And this is some sort of a way of getting around the fact that we don't really see any juveniles or adults. We can actually just look. We can take samples from their spine and get nice timelines from that, and do some isotope work on that as well. Again, anyone sees a dead whale shark, it'd be great if I could have some connective tissue, some red muscle tissue, some blood, and some liver samples, and maybe an eye, if you feel like throwing that in there as well. That'd be great. That work, like I mentioned earlier, would be really awesome as well, because it means I can start getting an idea of tissue turnover for these different sharks, and start getting an idea of actually what I'm looking at. It would be great. And I think that's it, pretty much. Thank you.